Chapter 14, Amniotic Fluid Analysis. Now, with the use of current ultrasound, amniocentesis is now a common and very safe procedure in obstetrics. The study of the fluid is performed for three main reasons. One, to enable any kind of antenatal diagnosis of genetic or congenital disorders early in fetal gestation. That means 15 to 18 weeks. Two, to assess fetal pulmonary maturity in later pregnancy in 32 to 42 weeks. And finally, to estimate and monitor the degree of anemia caused by isoimmunization or infection. Amniotic fluid is a liquid medium that bathes the fetus throughout gestation. The amnion is a membrane composed of a single layer of cuboidal epithelial cells which surrounds the fetus and is filled with this fluid. It protects the fetus while enabling it to move around, and it also plays an important role in biochemical processes. The cells and many components contain electrolytes, nitrogenous compounds, proteins, enzymes, lipids, and hormones. The dynamic of the fluid is very interesting and complex. Initially, it is produced by the amnion in the placenta, and its composition is similar to plasma. But as the gestation progresses, the fetus plays more of an active role in the composition, where water and solutes are exchanged between the fetus and its surrounding medium through different mechanisms, such as intestinal absorption after swallowing the amniotic fluid, capillary exchange in the pulmonary system as the alveoli of the lungs are bathed in the fluid, and fetal urination. So early in the gestation, a transudate passes through the skin and makes a contribution to the volume. Because of respiration in utero, the pulmonary surfactants produced by the cells mix with this and can be evaluated. In the later stages of pregnancy, fetal swallowing and urination play a major role in the volume and composition of the fluid. So as the fetus swallows the fluid, removing any water or electrolytes, replaces them through urination with metabolic products such as urea, creatinine, and uric acid. And then there's a similar exchange between the fluid and the maternal plasma. The maternal plasma would remove any waste products and then replenish the water, nutrients, and electrolytes. Now this is a very dynamic and changing equilibrium and happening every 2 to 3 hours. Now initially the volume increases as the pregnancy goes further, so approximately 25 to 50 mils are present at 12 weeks to about 800 to 1200 mils at 37 weeks gestation. The collection of removing amniotic fluid from the sac is called an amniocentesis. It is performed transabdominally or vaginally with simultaneous ultrasound examination. Now, it can be performed at 14 weeks gestation. However, the purpose of performing the procedure indicates when it's done. So, for example, if the amniocentesis is to detect any kind of neural tube defect or genetic abnormality, this is usually performed early in gestation at about 15 to 18 weeks, which allows enough time for the performance of the chromosome in chemical studies, which can include culturing any cells, as well as consideration of pregnancy termination if the fetus is determined to be abnormal at the parent's discretion. Later in pregnancy, it's primarily used to assess the pulmonary and overall health of the infant, and this is usually assessed at 32 to 42 weeks. The collection technique is an aseptic one, and it uses a very long sterile needle where the physician pierces through the abdominal and uteral walls with a long sterile needle and aspirates about 10 to 20 mLs of amniotic fluid into different syringes. These are numbered to prevent contamination for the entire collection, and after its collection, it is immediately transferred to the lab, carefully in a sterile plastic container. It also should be protected from light, especially if bilirubin will be tested because it breaks down with light present. During certain times, it may be necessary to determine if the fluid collected is in fact amniotic fluid, or if it was a mistake and it was aspirated from the bladder and collected as urine. And really physically, you can't distinguish between the two if it's urine or amniotic fluid, so this is where chemical constituents come into play. So amniotic fluid contains glucose and a significant amount of protein. And later on in pregnancy, the creatinine concentration would be similar to that of plasma. So in contrast, urine has essentially no protein or glucose and contains high concentrations of urea and creatinine. Let's discuss the physical examination of amniotic fluid. So it really should take place immediately upon receipt of the lab, and it consists of a visual assessment of color as well as turbidity. So normally the fluid is colorless or pale yellow. There's a distinctive yellow or amber coloration, which is associated due to the bilirubin that's present. A green color could indicate the presence of meconium. Now, meconium is a gelatinous or mucus-like material that forms in the fetal intestine as a result of swallowed amniotic fluid and fetal intestinal secretions. Biliverdin is responsible for a dark green color. Blood can also cause contamination and result in a pale to red kind of color. If blood is present, it should be centrifuged to remove any of the intact cells before hemolysis occurs, because this can interfere with the other tests being done. As for turbidity, usually there is some degree of turbidity depending on the stage of pregnancy. Early in pregnancy, there's very little particulate matter and it's not very turbid, but as the pregnancy progresses, you have increased amounts of fetal cells, hair, and vernix that sloughs off and remains suspended in the fluid. When there is premature delivery anticipated, or it's due to fetal distress or complications, we have to ensure the fetus will be viable outside of the mother's uterus. So to assess maturity and potential viability, we have to evaluate the status of the fetal lungs, and this is done using a fetal lung maturity test of the amniotic fluid when gestation is about 32 weeks. It's not performed any earlier because it will definitely indicate immaturity. So when we talk about respiratory distress syndrome, it is the most common cause of death in the newborn, and it's a primary concern definitely when a preterm delivery is necessary. It results from insufficient production of the surfactant at the surface in the lungs. So normally alveolar epithelial cells produce and secrete phospholipids in the form of something called laminar bodies. These lamellar bodies release their active surface compounds known as surfactants into this alveolar space. Surfactants are very important because they act to alter the surface tension of the alveoli, preventing the collapse during expiration or breathing out, and reduce the amount of pressure needed to reopen them when we breathe in. There has been a correlation discovered between fetal lung maturity and the gestational age of the fetus at the time of testing. So there has been a guide for clinicians when discussing the risks versus benefits in the decision of when to deliver this baby. So there is an approach to a mature result when using any of the fetal lung maturity tests. So there is a series of tests that can be performed until a mature result is obtained. That way they can safely remove the baby. So there is a rapid test such as a lamellar body count that should be performed, and in late pregnancy a rapid immunochemical test to detect phosphatidylglycerol. Additional testing is required when results are indeterminate or the cutoff value indicates immaturity. So you have to consistently do this series of cascade approach until you reach a mature status for safe removal. Now, in the fetal lungs, phospholipids are required to decrease that surface tension within the alveoli and preventing its collapse, as well as enabling gas exchange. So lecithin is a major pulmonary surfactant that performs this function. And sphingomyelin is a phospholipid found in numerous cell membranes. It's also present, but its functional role is not yet established. So until about 33 weeks gestation, the pulmonary system produces these two phospholipids in equal concentrations. But at 34 to 36 weeks, the concentration significantly increases for lecithin, whereas the sphingomyelin remains constant or decreases. Now, these have led to a calculation of a lecithin-sphingomyelin ratio, which has also become very important when evaluating fetal lung maturity or pulmonary status. So an LS ratio of less than 2 is associated with immaturity of the fetal pulmonary system, whereas 1 equal or greater than 2 indicates maturity. Phosphatidylglycerol is a phospholipid that also enhances the spread of surfactants across the alveolar surface. The unfortunate part is it's not normally detectable in amniotic fluid until 35 weeks gestation. A number of techniques have been used to identify this, and although the disadvantage would be you have to wait till 35 weeks of gestation, the positive effect of this test is if there are interfering substances, they're not affected by it, such as meconium in blood. The foam stability index, or shake test, is based on the physical and functional characteristics that surfactants play on amniotic fluid. So if adequate surfactants are present, a foam can be produced by shaking the fluid vigorously with ethanol, and the bubbles remain stable in the tube. Although this assessment is rapid and easy, sometimes it produces poor reproducibility, as well as contamination does affect these results. Now, fetal lung maturity can be indicated by a stability index of greater than or equal to 0.47. Lamellar body counts can also be used as another test for fetal lung maturity. Now, the pulmonary surfactants are stored in lamellar bodies. So the secretion of these bodies into the lumen of the alveolus begins at about 20 weeks gestation. By the third trimester, they are present in the amniotic fluid at concentrations ranging from 50,000 to 200,000 per microliter. and greater than 50,000 indicates that the fetal lung is mature. Again, contaminants can falsely elevate the count, but it is quick and it's very inexpensive, and only a small sample is needed. Normal gestation, bilirubin is not really detectable in amniotic fluid. But during normal red cell destruction in the fetus, the unconjugated bilirubin produced is removed rapidly by the placenta into the maternal circulation. Now, because the fetus has an immature liver, when there's a hemolytic disease process, it causes increased and persistent hemolysis of the fetus's red blood cells. So this production of unconjugated bilirubin increases significantly. So as a result, this increased amount of bilirubin enters the amniotic fluid, and it can be detected using a spectrophotometer. Now, hemolytic disease of the newborn occurs when maternal antibodies cross the placenta into the fetal circulation and destroy a large number of fetal red blood cells. Now, the amount of bilirubin in amniotic fluid is related to the severity of the hemolysis. So you have to do serial bilirubin measurements to monitor the condition. When normal amniotic fluid is scanned using a spectrophotometer, it ranges from 350 to 580 nanometers. The curve is obtained using a straight line that gradually decreases in absorbance between 365 and 550. Bilirubin has a maximum absorbance of 450, so as the concentration of bilirubin in amniotic fluid increases, the absorbance of the curve also increases. There is some kind of contamination issue with this process because the absorption of oxyhemoglobin is lower. So you will see a blip on the actual Lilly chart. Meconium can also cause a problem and is not acceptable for testing. Now, because we are testing bilirubin, it breaks down with light. These specimens must be protected from light after collection. They are very sensitive. So here's an example of a graph that is normally used, which it determines the fetal age to the severity of the hemolysis based on zones. So 1 is in a zone of normal fetal development. 2 distinguishes a marked type of hemolytic disease of the newborn, and 3 determines a severe hemolytic disease of the newborn. One can also use a Queenan chart to determine the risk of hemolysis. So values in the lowest zone indicate either an unaffected or mildly affected fetus. The intermediate and affected zones indicate an increasing severity of fetal anemia, and the uppermost zone indicates a severe hemolytic disease and a high risk for mortality of the infant.